All right. Hello, my name is Jordan Hess, and I am a master's student at the University of Texas at Dallas. And today we're going to be looking at how eating a very special type of yogurt may be able to help you reduce your stress. And to do that, what we're going to be looking at is the interaction between the gut, the immune system, and the brain. And we're going to be using an animal model of major depressive disorder known as chronic social defeat stress, or repeated social defeat stress. So for these chronic social defeat stress experiments, so we have the larger animal, right? So that's going to be this guy here. That's the retired breeder, the CD1 animals. And for the B6, the smaller black mice, those are going to be placed into the same cage with the retired breeder, and they will be bullied for 10 minutes each day for 10 days. Now, for each minute of those 10 minutes, there must be at least one 5 to 10 second bout. Okay, and then to capture some of the chronicity here, the animal will then be housed for the next 24 hours with only this perforated plexiglass divider separating them. Okay. Okay. And then afterwards, we want to measure how sociable the animal is after getting bullied, because what's going to happen is some of the animals, it will be like they weren't affected at all. They'll still, when put into a cage with a member of the same species, they'll go up to it and sniff it. Hello, how you doing? No problem, as if they were never beaten up. And some animals are really affected, and they will spend more time away from the stranger. And so here, if you think 10, 10, 10, so there's 10 minutes of habituation to that center chamber, and then 10 minutes that the animal is allowed to freely explore, and then the animal will be gently guided back to the center chamber, while a stranger of the same species will be placed in one of the plastic cages, and then an inanimate object will be placed in the other plastic cage. And then what the researchers are going to look at is the amount of time spent with the stranger versus the amount of time spent with the inanimate object. And they will also look at the time spent sniffing the stranger versus the inanimate object. Okay, and for that amount of time that's spent with the stranger as opposed to the inanimate object, that's going to be expressed here as a ratio, and it's called the social interaction ratio. And so for values less than 1, okay, that's going to be considered a stress susceptible animal. So the animals that are very impacted by being bullied, and then the animals that, it's like they never got bullied at all, that's going to be a social interaction ratio that's greater than or equal to 1. And if we look here, that makes sense, because as the animal spends less and less time with the inanimate object, okay, this denominator is getting smaller and smaller. The overall value is going to be getting larger, and that's what we're seeing here. All right, so as mentioned previously, the animals will undergo 10 days of chronic social defeat stress, followed by 24 hours of social interaction testing, and 24 hours after that, the tissue will be collected. And in previous experiments, researchers had identified interleukin-6 as a key mediator of the stress susceptible phenotype. And so here, what we're looking at, okay, we're looking at now instead that gut immune brain interaction, right? And so there's a couple different types of ways to measure diversity in the gut, and let's take a look. All right, and so for gut diversity, we have 3 different measures of gut diversity. So we have alpha diversity, beta diversity and gamma diversity. And for each of these, there's different subtypes. And here we're looking at Shannon alpha diversity. And so Shannon alpha diversity measures like the richness or the number of different taxa and the taxa evenness. And for the evenness, let's think like if we had, a more simple situation in which there was only 3 different taxa, okay? And in this case, let's say we had a perfectly even distribution. So say 33% of one, 33% of another, and type A and type B, and then 33% of type C. Or an uneven distribution like 10% of type A, 10% of type B, and 80% of type C. Well, the evenness is not the primary factor that's being captured here in this Y value, the Shannon index. And we can use the equation to get these numbers here, which is the approximate number of different taxa for each Y value. For a Y value of 5, that's 148 different taxa. For a Y value of 4, 54 different taxa. And for a Y value of 3, approximately 20 different taxa. And so when we look at the control in resilient conditions, we see that those are similar in terms of the Shannon alpha diversity, but for the stress susceptible animals, it seems that in some of those animals we're seeing compared to control, which has approximately 54 different taxa, here just over 20 different taxa. Okay, and for the beta diversity, this is going to be measuring intra-taxa variation, and this is a principal component analysis that has been done, which is to take many variables and reduce them into here 2 variables. And whereas before we were looking at a difference in the stress susceptible phenotype compared to the control and the resilient animals, here we're seeing an effect of, that's what I've highlighted here, is an effect of experiment. So for this particular measure of diversity, simply being bullied was having an effect, and this could be an area of interest for future investigation. Okay, so as far as like what type of bacteria were affected, well, it had been previously known that lactobacillus could have an effect on T cell populations, and that gamma delta T cells were able to affect behavior. And so this guided the researchers to look at Lactobacillus johnsonii. Now, so when the researchers looked at the relative abundance of Lactobacillus johnsonii, what they found was that in the stress susceptible animals, there was a reduced abundance, okay? And then they said, well, let's plot a correlation between the social interaction ratio in the presence of Lactobacillus johnsonii, and what they found is that there was a positive correlation such that the more that an individual falls into the category of, or an animal in this case, falls into the category of resilient, the more likely the animal is to have an abundance of Lactobacillus johnsonii. And so it's, in some sense, it seems that having this bacteria in the gut is conferring some form of resilience towards stress. Okay, and when we're talking about bacteria here, we have at the highest level genus, and then below that species, and below that strain. And so at the species level, we have Lactobacillus johnsonii, but the researchers wanted to know, like, okay, well, what about the different strains? And so if we look here, okay, and we look at these y-axis values and compare to these y-axis values as we go down from graph to graph, what we find is that these are the different strains listed from greatest abundance to least, and the LA1 is in the greatest abundance, which is going to be important later when the researchers are feeding the lactobacillus to the mice and observing the outcomes on sociability. Okay, and there's always a question of translatability, right? So these animal models that we're looking at, the chronic social defeat stress models, are supposed to be an animal model of major depressive disorder in humans. And so in terms of translatability, we want to know, is what we're doing with these animals, is it going to be relatable to humans? And so Here on the x-axis, we have some measures of fitness for how well does an individual fall into the category of major depressive disorder. And what is observed here is that there's a negative correlation such that the better that an individual fits into the category of major depressive disorder, the less likely it is for them to have an abundance of the lactobacillus, and that the MADRS was the strongest indicator for this. Okay, so previously I mentioned that the background suggested that there was a relationship between lactobacillus and T cell populations. And so here, what the researchers did is said, okay, well, so flow cytometry is a process used to look at cell surface markers. And so you'll use a fluorescence conjugated antibody, shine some light and look at the light that's returned. And here, so for the x-axis, we have FSC, which is the forward scatter. So this is a measure of the diameter of the cell, where the SSC is a measure of like the granularity or internal complexity or colloquially density of the cell. And here, CD3 is cluster of differentiation 3 is a marker that's telling us that we're looking at T cell populations. And then they said, well, now let's look at a subtype of T cell populations called gamma delta T cells. And what they found is that there was a greater abundance of gamma delta T cells in the stress susceptible condition. And so this is like basically produced in the gamma delta 17 T cells that produce this interleukin 17. Now, interleukin 17 is not a cell surface marker, so an additional step of permeabilization had to take place here. And so what they found is that not only was there a greater abundance of gamma delta T cells, but there was a differentiation into the gamma delta 17 producing subtype. Okay, and it's always important to confirm the findings on multiple levels. And here we're looking at some tissue. And in the upper panel, we have the lamina propria of the colon, Latin for proper plate. So inside the colon, that initial first, so inside the colon, the initial layer of cells, that's going to be the epithelium, and on the other side of that is going to be the lamina propria. Now, what we can see here, so the blues indicating nuclear material, and this green fluorescence is indicating the gamma delta T cells, and we see indeed a greater abundance in the susceptible animals. And when we look in the meninges, okay, we see that this is also true, okay, and that laminin is a meningeal marker just telling us that we are in the right place, and this is just an inset, okay, of this area here. And let's see what we have here. Okay, so now the researchers are going to look at if we feed the animals the LA1 strain, okay, what effects does this have on gamma delta T cell populations, and is there a effect on sociability? And so the researchers fed the animals the LA1 strain for 10 days prior to and during the chronic social defeat stress. And what they found is that there was no inherent effect of feeding the animals the LA1 strain on the gamma delta T cell populations, okay? But in the presence of the chronic social defeat stress, we no longer are seeing this increased abundance of gamma delta T cells, and the same is true for the differentiation into the gamma delta 17 producing subtype. And so this is an important point not to gloss over, because let's say that people were, like these results did translate into humans and people were able to eat some fancy yogurt to help them manage stress. Well, if individuals that didn't need it ate the yogurt, would it negatively affect their T cell populations or gamma delta T cell populations? And what this might indicate is no, though it could have other effects. But as far as the gamma delta T cell population, so it might be like safe for you to eat the yogurt, whether you fall into the category of stress susceptible or resilient. Okay, so that's an interesting point. And then as far as effects on sociability, so first we want to consider what the expected result is. And so let's look here in this vehicle condition. And for the chronic social defeat stress, notice that this isn't being teased out here into stress susceptible and resilient as it is in some cases. And so because of that, there's going to be intra-group variability. So in each cohort, there's going to be a different proportion of stress susceptible and resilient animals. And so because of that, what we're going to see is one of two things. So typically, basically what we see is that the animals that, have never been bullied or anything, they like to spend time with the other animal when presented the opportunity. So they're generally sociable creatures. And once they get bullied though, okay, so they tend to be less sociable, and so we'll either see those bars at a fairly even level, which is what's observed here, or we'll see that the time spent with the inanimate object, okay, this white bar will be higher. Okay, so those are the two expected outcomes. And so what the researchers find is that when they feed the animals the LA1 strain of the Lactobacillus johnsonii, there's not any inherent effect, but in the presence of stress, we see a suppression of the stress susceptible phenotype. And that's what's okay, so that's also being shown here, and I don't include these graphs because they're measuring the same thing, and so approximately. And so they're looking at the time spent in each chamber versus the time spent sniffing. Okay, and so here we see that if you give them if you feed them the bacteria, then you find that there is the animals are still sociable here. Okay. And here, this is just a heat map representing that when the animals are bullied, they tend to spend more time with the inanimate object, and that when you bully them but also feed them the LA1 strain, this difference goes away and they start spending more time with the stranger. Okay, so for T cells, for these gamma delta T cells to be exerting some behavioral effect, you might want them to be pretty close to the brain. And so the meninges is close to the brain, okay? And then so for those gamma delta T cells, they express that gamma delta T cell receptor on the surface, and there's the two arms, right? The gamma arm and the delta arm. And so for the gamma arm, we have a V6 and a V6 and a V7 subtype. And so for that V gamma 6 subtype, that's going to be found in greater abundance in the meninges, and the V gamma 7 subtype will be found in a lower abundance in the meninges, and the opposite will be true in the colon. Okay, and so when we look here, so the ND just means not determined, and so for the parts that are determined, we see that in the control in the resilient animals, there's an equal amount of the V gamma 6 subtype in the meninges, and that there's a small presence of V gamma 7. But in the stress susceptible animals, what we see is this expansion of the V gamma 7 subtype, but that's what's found in the colon. So possibly there's some migration from the colon to the meninges, and that's what the researchers are going to explore using antibodies. But first, we're going to look at an additional result that was achieved through the use of the antibodies. Okay, so here we have an antibody for the gamma delta T cell receptor, and we also have the isotype control immunoglobulin G, which is just the untrained antibody. Okay, and so the animals will undergo, as typical, the 10 days of chronic social defeat stress, and this will be followed by open field test, the extended plus maze, both of these being measures of anxiety, social interaction testing, and then a sucrose preference test. And let's take a look at this sucrose preference test over here on the right. So normally the animals will tend to prefer sugar water, and once they get bullied, they'll prefer it less, okay? And so this like reduced response to something that's typically pleasurable we refer to as anhedonia. Now, when the antibodies are administered prior to the chronic social defeat stress, we do not see the anhedonia. So that means that the mechanism that is responsible for the anhedonia is mediated, at least in part, by the gamma delta T cells. Okay, and so for this particular measure of anxiety here, as you can see here, there's a difference initially. And so what they're looking at is, so there's going to be this kind of plus looking structure with 2 closed arms and 2 open arms, and the amount of time spent in the open arms will be a measure of anxiety. And so the animals are still spending less time in the open arms. And what that means is that for this particular measure of anxiety, it appears to be independent of the gamma delta T cells. And so that's interesting, right? That the social defeat initiates like 2 different types of anxiety, one of which is mediated by the gamma delta T cells and the other which is not. Okay, and so this here is perhaps the most difficult image to interpret, the most difficult result to interpret. And so let's kind of break it down part by part. So in this left panel, we're in the lamina propria of the colon. In the right panel, we're in the meninges. Okay, and then in this upper part, that's when they receive the untrained antibody. And in the lower panel, that's when they receive the trained antibody. And overall, this is that flow cytometry where we're looking for the presence of the gamma delta T cell receptor. And the same is true here. So we have the untrained antibody, the trained antibody, and we're looking at the gamma delta T cell receptor. So this antibody is being administered intraperitoneally to the gut, okay? And what we see is an ability of the antibody to act locally. Okay, so there's a reduction here from 9.2% to 2.9%. But when we look at the meninges, we see that there is no meningeal reach of this antibody. Okay, and so what we would expect then, okay, is that for this number here, 24.5%, to be pretty close to this number, but it's not. It's reduced. And so what that tells us is that, okay, so these cells are typically migrating to the meninges, but there's no migration that occurs. No migration occurs here because of the reduction that happens locally when you administer the antibody. Okay, and then of course the researchers wanted to look at the effects on sociability, and they saw no inherent effects, but under stress they saw suppression of the stress susceptible phenotype, and that is also indicated here in the heat maps. So typically they're spending more time with the inanimate objects when they're bullied, and that when they receive the antibody they're spending time with the stranger animal again. And so here what we have, okay, they said, you know, because you could get rid of the gamma delta T cells to some degree with the antibody, but if you want to remove them absolutely, you could do a knockout. That's what this T cell receptor delta knockout animals are. So these animals are going to be deficient in the gamma delta T cells. And then we have this idea, you know, of adoption where you can take cells from a healthy animal and transplant them into the animals that are deficient. And so you might want to know how well those cells are taking to the host, which we refer to as engraftment. And specifically, are they, you know, differentiating into the IL-17 producing subtype? And so to know this, the researchers put an indicator in the gene such that when it's turned on, it will produce green fluorescent protein. So these gamma delta T cells are adopted 1 day before and 5 days after beginning the chronic social defeat stress. Okay, and it's important to note here that all these animals are deficient in gamma delta T cells. And so in this vehicle condition, what we see is that being deficient in gamma delta T cells doesn't have an inherent effect on sociability, but under chronic social defeat stress conditions, we see a suppression of the stress susceptible phenotype. And adopting the gamma delta T cells, okay, again, doesn't have any inherent effect on sociability, but in the presence of chronic social defeat stress, we see an emergence of the stress susceptible phenotype. And as far as like how well are those cells taking to the host, so here what we're looking at is the IL-17 producing subtype of gamma delta T cells, and that's indicated here in the lamina propria of the colon, and here in the meninges as being in a greater abundance in the chronic social defeat stress conditions. And so for Dectin-1, so Dectin-1 is what we call a pattern recognition receptor, okay? So here we have a pathogen, okay? And so on the surface of that pathogen, there's going to be pathogen-associated molecular patterns, which are just geometrical features on the surface of the pathogen. And so there's three kind of different methods of identification of the pathogen based on those geometrical features. And so it could be through the serum or tissue fluid, through a receptor on the membrane, or a receptor in the cytoplasm. So in the serum, we refer to these pattern recognition receptors as soluble, and we refer to them as opsonins. And so they will coat the microbe in a process known as opsonization, okay, or microorganism in a process known as opsonization. And then through a process known as opsonic recognition, will have an indirect identification of that pathogen, whereas here we have with the membrane-bound pattern recognition receptor, we have a method of direct recognition, okay? And so that's what that Dectin-1 is, okay, is a membrane-bound pattern recognition receptor, and it binds beta-glucans, which are found in the cell wall of fungi. So this is part of the body's innate immune system, and it just so happens that Dectin-1 was previously shown to be able to impact T cell populations. And so the first thing that the researchers did was to look at the presence of Dectin-1 expressed on the gamma delta T cells, and that what they found is that there was a greater abundance of Dectin-1 in the stress susceptible animals, okay, as is indicated. And then the researchers said, well, okay, we know that the gamma delta T cells are expanding, right, and differentiating into the IL-17 producing subtype, but to what degree, under stress conditions in some animals, but to what degree is this being mediated by Dectin-1? And so what they find is that this stress-related expansion and differentiation into the IL-17 producing subtype is being mediated by Dectin-1, and that we see the expected results on sociability in that there's the animals are still sociable, okay, that relative to the controls. And so for Pachyman's a beta glucan, and so it's going to be acting on Dectin-1. And so this is kind of verification where they're going to look at, the effects on gamma delta T cell populations and the effects on sociability, but they're also kind of wanting to verify that it is binding to where they think that it's binding, right? And so it should be binding to gamma delta T cells, right? So Dectin-1 on the surface of gamma delta T cells. And so here where we see all these arrows, this is a co-occurrence of Pachyman and gamma delta T cells, indicating just that. And when the Dectin-1's knocked out, we see less arrows, okay, indicating that the Pachyman is indeed binding to Dectin-1 on the surface of gamma delta T cells. And so when we look at the effects on gamma delta T cell expansion and differentiation, what we find is a very similar effect to knocking out Dectin-1, okay? And then this is also true when we look at the effects on sociability. So the animals, again, are still sociable relative to the controls. And to kind of combine some techniques together, so when you're adopting cells from a wild type animal to an animal that's deficient in those cells, you could adopt the wild type cells if you wanted, but you could also adopt the some cells that have been modified, say if you wanted to knock out Dectin-1 on those gamma delta T cells. And so what they did is just that. And what they found is that there was a reduced engraftment and differentiation into the IL-17 producing subtype in the Dectin-1 knockout animals. And so that kind of tells us that Dectin-1 on these gamma delta T cells under stress is mediating a couple different things in the susceptible animals, and it's mediating the engraftment, and it's mediating a differentiation and expansion of those gamma delta T cells into the gamma delta 17 producing subtype. And so there's a couple of questions here that we might want to ask, and one of them would be like, well, how are the gamma delta 17 T cells in the meninges exerting their effects on behavior? Is it through the interleukin 17? And if so, what are the next steps in that mechanism? And previously, researchers had identified interleukin 6 is a key mediator of the stress susceptible phenotype. And so interleukin 6 wasn't actually discussed in this paper, so that would be a good area for future direction. And then an additional area for future direction kind of asked, well, why in the stress susceptible animals is that particular population, Lactobacillus johnsonii, susceptible, and why do we find it in decreased abundance? And so just to rehash what we looked at here, so in the stress susceptible animals, so if we look on the gamma delta T cells, Dectin-1's expressed on the surface. Now, the Lactobacillus johnsonii populations are going to decrease. Okay, when those populations decrease, then we're going to see through Dectin-1 an expansion of the gamma delta T cells and differentiation into the gamma delta 17 producing subtype. Then there will be peripheral from the colon to meningeal migration of these gamma delta T cells to exert their behavioral effects. And so there's this idea that, maybe what holds true in the mice will hold true to some degree in humans. And what that means for us is that one day we may be eating some very special yogurt to reduce our stress. All right, I hope that you enjoyed the video. If you did, be sure to like and subscribe and leave any comments or questions that you have below. Again, my name is Jordan Hess, and I look forward to seeing you in the future.